Bismillahirrahmanirrahim. Today, inshallah, we will complete our survey about meiosis. I've talked about this in our last video. We shall elaborate on some of the details on this today, inshallah. As I've suggested before, mitosis is a process that occurs in somatic cells or body cells and starts with a diploid cell and results into identical diploid daughter cells. Whereas meiosis occurs in gamete producing cells where the mother cell, parent cell, is a diploid cell. In humans is 46 chromosomes, and the end results are 4 haploid cells with the half of the genetic material. And these are gametes, and in humans, sperm and eggs. And the process of mitosis, of course, occurs in somatic or body cells. We had also previously reviewed the phases of mitosis, PMAT, prophase, metaphase, anaphase, and telophase, where in prophase the duplicated sister chromosomes come together and start to line up in the middle, and the spindle fibers are attached. And in metaphase, the chromosomes line up in the middle and in the metaphyseal plate. And in anaphase, they're split apart, and telophase completes the process of cell division by pinching of the two cells like that. We have suggested a very important distinction between mitosis and meiosis occurs in metaphase 1, the first metaphase, where the two homologous chromosome pairs line up in the middle, forming that tetrad. And that tetrad is very critical in this process. So let me try to suggest to you again that process. So this is meiosis 1, the first part of meiosis, where you have the formation of these tetrads. Oops, sorry, like these over here. All right. So the tetrad also allows of crossing over of chromosomes. Okay. Pardon me for the interruptions here, but I just want to make sure you understand. So observe here the formation of the tetrad and separation of the homologous chromosome that occurs in metaphase of meiosis 1. And then these two, the first two cells that are formed are two different cells, and when they do decide to form the 4 daughter cells, and the end result is eventually creation of 4 different cells. If you notice over here, none of these cells are identical due to crossing over chromosomes, and that occurs in metaphase. Now here's again the same process demonstrated. Formation of tetrad, crossing over, separation of the homologous chromosomes, and then the second division, which is very similar to mitosis, where the chromosomes line up in the metaphyseal plate and the two sister chromatids are separated, producing 4 daughter cells. So this is a process that's beautiful and important to understand, that results in formation of 4 haploid cells. Okay. Centrioles. Centrioles are quite remarkable structures. I hope you get an opportunity in life to actually study these, and they're quite beautiful. Now, I just wanted to suggest this, okay? So this is in metaphase 1 of meiosis that occurs where you form those tetrads, okay? Now, you have to understand, if you have these, in this case, in this example, you have 3 pairs of tetrads that occur. Now, nobody says they have to line up in a certain way, right? So they can line up, for example, let's say blue is from dad and red is from mom, as paternal and maternal chromosomes. They can line up all blue and all red like this, or blue, blue, red, and like this, or various different combinations. So as you can see, the individual chromosomes can line up in numerous different ways. Okay, and this results in also creation of variability in the gametes that are produced. For this reason, if they line up like this, the gametes that are produced are going to be different than, let's say, if they were to line up like this, the gametes will be different. So depending on which way these line up, okay, it will determine which gametes will have which chromosomes from which dad. In other words, if, like, we have, for example, 43 chromosomes, right? Now, this is only an example of 3 of them, right? So imagine if you have if you have like 43 chromosomes, right? Or 23 pairs of them, so all lined up. The various different ways that they can line up gives to approximately over 1 million different combinations that can occur. In other words, a particular individual from one of these cell divisions could produce up to that many different types of gametes. So you can do some probability math to figure that out, but there are an innumerable number of different combinations of these. And add to that the crossing over phenomenon, okay, which adds further to genetic variability. This is the reason why if parents were to have, let's say, 1 million kids, they will all look different. Now, here's again emphasis on the same phenomenon. This is a cell that has only 2 chromosomes, and this is paternal. It's just for the sake of studying this. The blue is dad, red is mom. Dad's chromosomes, mom's chromosomes are duplicated, right? And then if they line up like this, okay, the daughter cells produce in the first division will be like this, and then when they divide, it gives you these 4 gametes. But if they happen to line up in this way, and when they separate, they'll give you these 2 cells over here, and then when these 2 divide, and they'll give you these 4 combination of gametes or daughter cells. So as you can see, there's a variability that results as the way these are lined up. This phenomenon is called law of independent assortment. It's another actually Mendelian hypothesis and theory. He said the genes must be able to separate into gametes independently, and for example, this chromosome has no power over these. In other words, it doesn't dictate which way these 2 line up, and these don't dictate which way they line up. They could equally line up like this, and they could equally line up like this. One does not affect the other in terms of how they line up. This is called law of independent assortment. It's like equal opportunity lineup, so to speak. So now consider this then. If you have the human karyotype of this 23 pairs of chromosomes, right, and altogether 46, and you are going to produce from this 4 gametes, right? If you're going to produce 4 gametes from these, now this could go over here, and this could go over here, and this could go over here, and this one could go over here, and this one could go over here, and etc. So, I mean, there are numerous different possibilities of how all of these will sort, as long as everybody gets, 23, 23, 23, 23. And depending on which way they line up. And this gives, as I said, an exceptional amount of variability in the gametes that are produced. As I said, and add to that this phenomenon of crossing over, whereas when you form that tetrad in metaphase 1 of meiosis, homologous chromosomes can cross over. Now, understand this, that it will be these 2 chromatids. Now, we have talked about this before. This terminology is quite interesting and confusing, and to give credit to biologists, and sometimes they come up with interesting terms. Now, these are 2 identical chromosomes. They're duplicated chromosomes, right? This one and this one, they're identical chromosomes that had duplicated themselves. Okay, individually this is called a chromosome, and individually this is also a chromosome. So it's like that's a chromosome, and the whole thing is also a chromosome. It's confusing, yes, but that's the way it is. Now, but this, you can call this one sister chromatids, okay? These are called sister chromatids, okay? And that tells you those are the ones that are hanging out together like that. So anyways, these are homologous chromosomes. That means this is dad's and this is mom's version chromosome, and they have duplicated themselves like this. And during the metaphase 1, when they line up in the middle, crossing over can occur, and it occurs in these 2, in the ones that are closer to each other, like this, right? So these are recombinant chromosomes over here, and the ones over here occurs in non-recombinant chromosomes that remain the same. So this results in creation of two different chromosomes, okay? Now this chromosome was identical to this chromosome over here, but no longer. See? A new chromosome is in fact created. Okay, so this crossing over phenomena is quite important in meiosis. The other thing that I just want to bring to your attention is the farther away genes are the more likely that they will be separated from each other. Okay, that's just because of simple anatomical restrictions, right? So if these are close together, more likely they will stick to each other, right? But if they're farther from each other, it's more likely that this guy will be separated from that guy. So the closer the genes are together to each other, the more likely that they will stay together. The farther away they are from each other, the more likely that they will be separated because of crossing over. Now, here's meiosis again, crossing over. And God bless the people who made these. I didn't create these, but they're so invaluable. May Allah bless them. Now, so this is meiosis, right? Meiosis doesn't always work properly. There can be problems with meiosis. One of the things that can happen is a phenomenon called nondisjunction. Nondisjunction means that these two fail to separate. Okay, whereas both of them, this one's supposed to get this one and this one, but it kind of dragged around that one along with it, and this one is missing that pair. So this is called nondisjunction. This is when meiosis goes awry. So watch what happens here. So nondisjunction can cause trouble. Now observe. If there's non-disjunction, for example, over here, resulting in these two, this cell will actually just missing a whole chromosome set, right? And this one will have extra. So one cell pair will have an extra, and one will have one less chromosome pair. Okay, so non-disjunction can cause one cell to have one extra chromosome pair and the other one to have one less. Non-disjunction can occur in the first part of meiosis or the second part of meiosis, part meiosis 1 or meiosis 2. So and the results are very different. If it occurs in meiosis 1, if non-disjunction occurs in meiosis 1, right over here when they're lined up as a tetrad in metaphase 1, the cells created will be like this. So all of these will go over here, then you have this, right? Okay, and when this cell divides like this, you'll have one cell that has one extra chromosome, another cell that has one extra chromosome. That's because from this extra one, and this is the daughter cell that's missing one. That's from here. It's missing one chromosome, and when that divides, you'll have two daughter cells that have one less. So if meiosis 1, if non-disjunction occurs in meiosis 1, you'll have two daughter cells that have one extra chromosome and two daughter cells that have one less chromosome. Now look at in meiosis 2. So look at a second scenario. Meiosis 1 here is fine. Okay, it works just fine over here. But non-disjunction occurred in meiosis 2. Whereas this chromosome did not, it was supposed to go over here, it didn't disconnect, and there was non-disjunction, so it retained this one. So it has one extra chromosome, and this one lost a chromosome. But since this cell was not affected by this, okay, so this problem occurred over here, this one is unaffected, so this will still produce two normal cells, okay? So it will produce two normal gametes. Okay, these are the normal gametes. So these are normal gametes. Okay, now so meiosis 2 will produce at least two normal. If the non-disjunction occurs in meiosis 2, it will produce at least two normal gametes. If non-disjunction occurs in meiosis 1, none of the gametes produced will be normal. Either they will have one extra chromosome or one less chromosome. And this does happen in real life. So this is an example of a non-disjunction that has occurred in what? Meiosis 1 or meiosis 2? See, none of these cells are normal. right? So this non-disjunction had to occur in meiosis 1, right? So this results in either one less chromosome or one extra chromosome. And this one is particularly talking about chromosome 21 in the human karyotype. Chromosome 21 is a syndrome. It's called Down syndrome, reasonably common. There are 3 copies of chromosome 21 in this individual. So here's the 2 chromosomes that resulted as a non-disjunction in the gamete. And then when that egg was or sperm fertilized the egg, then that it brought with the other chromosomes. So now there's a total of 3 chromosomes here. So Down syndrome is trisomy 21. These, you probably have seen these individuals. They tend to be very sweet people, nice people, happy people, although they can have different temperaments, and there are developmental delays and physical features that are characteristic of this individual, of these individuals. And so Down syndrome, or this is a trisomy 21, which results as a non-disjunction. Now this was described in this slide, if I could show it to you. Now watch what happens here. Non-disjunction, and if this one was fertilized, now there would be trisomy 21. See, there would be 3 over there. It's kind of fast, but if you keep your eyes there, and they're fertilized, now they have an extra pair. So this is how trisomy 21 occurs. Is a result of a non-disjunction in meiosis. And that concludes our chapter on genetics and meiosis. Until next time, assalatu wassalamu ala rabbil alameen. Assalamu alaikum.